Okay, so in the last class, we have completed the stoichiometry of the fatty acid biosynthesis. That is, what is the overall chemical reaction happening during the synthesis of a C16 palmitic acid, C16 saturated fatty acid, that is the palmitic acid, right? So the sources or the substrates required are uh, 8 molecules of 2 carbon acetyl CoA molecules, right? So 2 into 8, 16, right? But the acetyl CoA molecules cannot condense as such because it's a thermodynamically unfavorable reaction because huge amount of energy is required for a condensation between two covalent bonds or for two carbon atoms to undergo condensation. So what is the alternative? One of the acetyl CoA molecule will be coming in the form of acetyl CoA acid, but remaining all the 7 molecules are coming in the form of malonyl CoA, right? So the 7 molecules of malonyl CoA condenses with each other except for the first step. In the first step, one molecule of acetyl CoA condenses with malonyl CoA, giving rise to the higher molecule 4 carbon butyl CoA. Now the butyl CoA condenses with 2 carbon or malonyl CoA, next molecule of malonyl CoA to condense together, and it carries out 4 steps of reactions, and finally it gives rise to 6 carbon, and hence the cycle is proceeded till 16 carbon palmitic acid is formed. And the whole reaction is catalyzed by a multi-enzyme complex we known as which is known as fatty acid synthase complex, etc. So a complex, etc. So one molecule of acetyl CoA condenses with 7 molecules of malonyl CoA in presence of 14 molecules of NADPH, right, giving rise to 1 molecule of palmitic acid. That is, the reaction is catalyzed by 1 molecule of fatty acid synthase എന്ന കാര്യമാണ് പറയുന്നത്. പക്ഷേ we know that fatty acid synthase complex is a dimer. Hence, at a time, we can have 2 molecules of palmitic acid. That is, 2 sets of reactions are happening at a time. Clear? So, at the end of 7 cycles, we will be having 2 molecules of palmitic acid. That is, a total of 16 molecules of acetyl CoA reacts together, right, in a side-by-side manner to give rise to 2 molecules of palmitic acid or palmitoyl CoA, right? So that's how the reaction is happening, consuming 14 molecules of NADPH, giving rise to carbon dioxide, water, then palmitic acid, etc. Right, so the stoichiometry was over. Then we have discussed the regulation of fatty acid biosynthesis. Usually, the reaction should be regulated by fatty acid synthase complex, but it's not this. It's not case in the case of fatty acid synthesis. Here, the fatty acid biosynthesis is under control of another enzyme which is known as acetyl CoA carboxylase, which is another multi-enzyme complex, and this is the deciding factor whether the fatty acid biosynthesis has to be taken place or not. Clear? So because once a malonyl CoA is formed as from acetyl CoA as a result of carboxylation, not decarboxylation, carboxylation reaction, the malonyl CoA is committed to enter into fatty acid biosynthesis, right? So it thinks twice for the synthesis to happen. അതുകൊണ്ട് തന്നെ the rate limiting enzyme is the acetyl CoA carboxylase enzyme, right? So in presence of acetyl CoA carboxylase, the acetyl CoA molecules are converted into malonyl CoA molecules, and then the fatty acid synthase comes into action and carries out the biosynthetic reaction, right? So that's the rate limiting step. And again, the acetyl CoA carboxylase is regulated under 2 mechanisms. One by means of allosteric regulation, where the allosteric modifiers will be there. Their citrate is an inhibitor. Then PFK1 can act as an inhibitor, right? Then well-fed state, only in the well-fed state, that is carbohydrates, proteins ഒക്കെയാണ് അല്ലെങ്കിൽ surplus amounts of carbohydrates and proteins അവരൊക്കെ അതുപോലെ are acting as activators, while the product fatty acid CoA will be acting as an inhibitor, right? By means of feedback inhibition. So there are different types of activators and inhibitors. That is how the allosteric mechanism is carried out. There is a second mechanism which is a covalent modification, which is controlled by hormonal regulation, and the main hormones involved are the glucagon and insulin. Glucagon always suppresses the or inhibits the fatty acid biosynthesis, while insulin enhances the fatty acid biosynthesis, and glucagon is activating the fatty acid synthesis by means of activating another enzyme, AMP kinase, which carries out the activation of AMP kinase. This AMP kinase carries out the phosphorylation of acetyl-CoA carboxylase, converting it from the active state to the inactive form. That is, acetyl-CoA carboxylase, when undergoing phosphorylation, will be converted into the inactive form. And this phosphorylation is carried out by glucagons, as a result of which the fatty acid biosynthesis will be inhibited. Clear? So the role of glucagon is to prevent the fatty acid biosynthesis, or it inhibits the fatty acid biosynthesis, or it inhibits the acetyl-CoA carboxylase by converting it into the phosphorylated form. Clear? While insulin carries out the function of the opposite reaction, opposite mode. That is, it dephosphorylates the acetyl-CoA carboxylase and activates the fatty acid biosynthesis. Right? So these are the major hormones which are coming into action. So all these mechanisms comes under short-term regulation. That is, in between meals, for a short period of time. അവിടെ മാത്രമേ role ഉള്ളൂ. പക്ഷേ long-term regulations also are carried out in the case of fatty acid biosynthesis, and the long-term regulation is under the control of genetic mechanism. That is, the enzyme is controlled at the genetic level, right? The genes which are regulating, which are responsible for synthesizing acetyl-CoA carboxylase is undergoing activation or inhibition. So that's a long-term phenomenon, right? So these are the two methods with which the rate-limiting enzymes are undergoing regulation, right? Now next is what happens to the palmitic acid formed in the cytosol? What's the fate of the fatty acid synthesis, or fatty acid CoA thus formed? What is happening to palmitic acid then? So usually C16 palmitic acid will be formed and will be stored. It will be present in the cytoplasm. But C16 palmitic acid, of course, it will be carrying out various mechanisms. But the body is in need of very long term, very long chain fatty acids, especially in the case of brain cells, etc. Brain cells, very long chain fatty acids are unsaturated, polyunsaturated fatty acids. So how can it be synthesized? So how can all these unsaturated fatty acids and very long chain fatty acids all are getting synthesized from the palmitic acid itself, right? So how is it carried out? And it is again carried out by elongation. നമ്മൾ ബ്രേക്ക് ഡൗണിന്റെ കേസിൽ വെരി ലോങ് ചെയിൻ ഫാറ്റി ആസിഡ്സിനെ സ്മോൾ ആക്കിയിട്ടാണ് അല്ലെങ്കിൽ ലോങ് ചെയിൻ ആക്കിയിട്ടാണ് ദേ ആർ അണ്ടർഗോയിങ് ഓക്സിഡേഷൻ. ഇനി സിന്തസിസിന്റെ സമയത്ത് ആദ്യം ലോങ് ചെയിൻ ഫാറ്റി ആസിഡ്സ് ആർ ഫോം, that is C16 fatty acid, C16 or C18, right? അല്ലെങ്കിൽ C16 ആണ് ഉണ്ടാവുക. C16 palmitic acid ആണ് എല്ലാ ടിഷ്യൂസിലും ഉണ്ടാവുക, except in the case of lactating mammary glands. Lactating mammary glands the synthesis will be stopped once short chain fatty acids are formed. Or C6 to C8 വരെ ആയി കഴിഞ്ഞ് കഴിഞ്ഞാൽ പിന്നെ സൈക്കിൾ അവിടെ സ്റ്റോപ്പ് ചെയ്യും. ഇറ്റ് വിൽ നോട്ട് ബി എക്സ്റ്റെൻഡിങ് അപ് ടു ദ സിന്തസിസ് ഓഫ് palmitic acid. Palmitic acid ഒന്നും ഫോം ചെയ്യില്ല, ബിക്കോസ് ലാക്ടേറ്റിംഗ് മെമ്പറിൻ ഓർ മിൽക്ക്, ബ്രസ്റ്റ് മിൽക്ക് കണ്ടെയ്ൻസ് ഓൺലി ഷോർട്ട് ചെയിൻ ഫാറ്റി ആസിഡ്സ്. അപ്പൊ C6 to C8 ആയി കഴിഞ്ഞാൽ ദ സൈക്കിൾ വിൽ ബി സ്റ്റോപ്പ്. അപ്പൊ C6 ഫാറ്റി ആസിഡ് ഉണ്ടാവണമെന്നുണ്ടെങ്കിൽ 5 cycles ഓഫ് സിന്തറ്റിക് പാത്വേ നടക്കും, അതിനുശേഷം അത് സ്റ്റോപ്പ് ആവും. അല്ലെങ്കിൽ C8 ആണ് വേണ്ടതെന്നുണ്ടെങ്കിൽ 7 cycles നടക്കും, എന്നിട്ട് അവിടെ നോട്ട് 7 cycles, 3 cycles അവിടെ നടക്കും, എന്നിട്ട് അത് അവിടെ സ്റ്റോപ്പ്. Clear? So that is how the reaction is happening, right? But except for the lactating mammary glands, other tissues, all the other tissues ends up in the formation of palmitic acid. എല്ലായിടത്തും palmitic acid ആണ് ഫോം ചെയ്യുക. And once the palmitic acid is formed, how is it elongated? The fatty acid synthase complex, the multi-enzyme complex fatty acid synthase cannot carry out the elongation process. It is not able to carry out the chain elongation. So chain elongation is carried out at an entirely different site. It's not happening in the cytosol, or the enzyme required for chain elongation is not present in the cytosol. So it has to be taken to somewhere else. So where is the chain elongation happening? It is happening in the microsomes or smooth endoplasmic reticulum. So microsomal chain elongation. So there are 3 systems of fatty acid biosynthesis. The extramitochondrial system is there, which ends up in the formation of C16 fatty acid, that is a palmitic acid. Once the palmitic acid is formed, then the elongation phenomenon is happening in by another mechanism which is known as microsomal chain elongation. So for that, this cytosolic palmitic acid will move slowly towards the microsomes, and in the microsome there is elongate system. Our chain elongation carry out ചെയ്യാൻ വേണ്ടിയിട്ടുള്ള enzymes ഉണ്ട്. അപ്പോൾ elongation എന്ന് പറഞ്ഞാൽ again suppose ഇപ്പൊ നമുക്ക് C16 ഉണ്ടായി, നമുക്ക് വേണ്ട ഒരു C22 fatty acid ആണെന്ന് വിചാരിക്കുക. So it's a very long chain fatty acid. അല്ലെങ്കിൽ C18 stearic acid ഒക്കെയാണ് ഉണ്ടാവേണ്ടത് എന്നുണ്ടെങ്കിൽ what is to be done? We carry out elongation, right? The same reactions are happening. That is, condensation happens, followed by reduction. Reduction is followed by again dehydration. Again, there's one more reduction. പക്ഷേ the enzyme is not fatty acid synthase. It is the microsomal chain elongase system, right? That is, microsomes contain, the microsomes of the smooth endoplasmic reticulum contains another enzyme which is carrying out the fatty acid chain elongation, which carries out all the same reactions which has been carried out by fatty acid synthase in the cytoplasm. Cytoplasm FAS എന്താണോ ചെയ്തത്, that is in the form of multi-enzyme complex. അവിടെ എന്തായിരുന്നു? It is happening, carried out by E1 to E7 with the help of acyl carrier protein. ACP എന്നുള്ള ഒരു carrier protein. പാൻ എസ് ആണ് ഒന്നിൽ നിന്ന് വേറെ ACP ആണ് carry ചെയ്ത് കൊണ്ട് അവസാനം പാൻ എസ് മേളിൽ ബ്യൂട്രൈൽ കോയിനെയൊക്കെ കൊണ്ടുവച്ചത് അല്ലെ. അപ്പൊ ACP is a carrier protein there. പക്ഷെ the same reactions are happening in the case of microsomes also, in order to convert a C16 fatty acid to C18 fatty acid. അപ്പൊ C16 രണ്ട് കാർബൺ ആഡ് ചെയ്താലേ എന്താവുള്ളൂ? C18 ആവുള്ളൂ. സോ അഗൈൻ എന്തൊക്കെ നടക്കണം? ഈ കണ്ടൻസേഷൻ, റിഡക്ഷൻ, ഡീഹൈഡ്രേഷൻ, അഗൈൻ the second round of reduction has to happen. പക്ഷേ it is not the fatty acid synthase which is carrying out the chain elongation, because the reaction is happening in an entirely different site. It is in the microsomes. But the same reactions are happening, but it's not catalyzed by fatty acid synthase complex. Allengil all the reactions are carried out by the same enzyme, but these enzymes are not present in the form of a multi-enzyme complex. Or a multi-enzyme complex, they are present in the form of individual enzymes. That is, condensation product comes out, then it will be acted upon by reductase enzyme in presence of NADPH. The reduced 3-beta or beta keto fatty acyl CoA will be formed, which is again undergoing dehydration by dehydratase enzyme, right, which is again followed by the second round of reduction, right? But so all the enzymes are present in the form of individual enzymes, right? Again, another difference is that there is no അതുകൊണ്ട് തന്നെ individual ആയതുകൊണ്ട് തന്നെ അവിടെ എന്താ ആവശ്യമില്ല? carrier protein ആവശ്യമില്ല. So that is, ACP is absent in case of microsomes. So the substrate is again malonyl CoA. നമ്മുടെ fatty acid synthesisലെ cytosolic processൽ എന്താണ് ഉണ്ടായ? extra mitochondrial systemത്തിൽ the substrate was malonyl CoA. The same malonyl CoA is again entering into the chain elongation also. Chain elongation, അവിടെ കണ്ടൻസേഷൻ നടക്കണം. അപ്പൊ രണ്ട് അസിറ്റൈൽ CoA ഒരിക്കലും കണ്ടൻസ് ചെയ്യില്ല. അതുകൊണ്ട് തന്നെ C16 palmitic acidലേക്ക് വന്ന് അറ്റാച്ച് ചെയ്യണത് എപ്പോഴും acetyl CoA is coming in the form of malonyl CoA. പക്ഷേ there is no multi-enzyme complex acting for carrying out the chain elongation. It's carried out by the microsomal chain elongase system, right? And in all the tissues, especially in case of liver, അതുപോലെ in the case of adipose tissue, അവിടെയൊക്കെയാണ് the active synthesis നടക്കുന്നത്, elongation നടക്കുന്നത്. Again in the brain cells also. ബ്രെയിനിലും elongation, because ബ്രെയിനിൽ സ്ഫിംഗോലിപ്പിഡ്സ് ഒക്കെ ഫോർമേഷൻ അവിടെ നടക്കണം, polyunsaturated fatty acids ഒക്കെ നടക്കണം. അതുകൊണ്ട് elongation എപ്പോഴും നടക്കേണ്ട ഒരു സൈറ്റ് ആണ് brain, liver, adipose tissue. So the chain elongation is actively happening, of course, in all the tissues, especially in case of liver, adipose tissue, and the brain cells. Right, so the electron donor is again NADPH, and the substrate is also malonyl CoA. But the difference is that there is no presence of carrier protein. അതായത് acyl groups നെ carry ചെയ്യാനായിട്ട് ACP യുടെ ആവശ്യമില്ല, because it's not a multi-enzyme complex. Clear? So that's the only difference. Otherwise, all the reactions are the same. So എന്തൊക്കെ ഉണ്ടാവും? ഇവിടെ malonyl CoA വരും. അതായത് acetyl CoA undergoes carboxylation. So the acyl group comes in the form of malonyl CoA. It condenses with the palmitoyl CoA. So malonyl CoA and palmitoyl CoA condenses together to give beta keto stearoyl CoA. That is, add ചെയ്ത് കഴിഞ്ഞു with the release of a carbon dioxide. Extra and carbon dioxide അങ്ങോട്ട് പോയി. Condensation ആണ്. അപ്പം condensation will be followed by the release of carbon dioxide. Carbon dioxide പോയി. 2 carbon will be getting attached. C18 fatty acid നമ്മൾ വിളിക്കുന്ന പേര് stearic acid എന്നാണ്. അപ്പം the product after the formation of condensation is known as 3-keto stearoyl CoA, which is followed by reduction carried out by NADPH. Reductase enzyme varum and converts the 3-keto to or beta keto to 3-hydroxy or beta hydroxystearoyl CoA. Beta hydroxystearoyl CoA is undergoing dehydration to give delta to trans octadecaenoyl CoA, then followed by second round of reduction reaction to give rise to what? Stearoyl CoA. Clear? So that is how the elongation is taking place. Even though I'm not having a slide over here, for carrying for showing the elongation, I've explained in detail in my notes. Okay, so go through it, write down the notes. So that's how the elongation is happening in case of the tissues, right? Once a palmitic acid is formed, the palmitic acid undergoes chain elongation by means of microsomal chain elongase system. That's the second system where the chain elongation happens with the help of all the same enzyme but not existing in the form of a multi-enzyme complex, and there's a complete